Hello there, welcome. Dr. Ken here, and today we will be going over the diffusion, osmosis, and enzymes lab. Okay, molecules have a type of energy called thermal energy due to their constant motion, and diffusion is the tendency of particles to spread out evenly in an available space. The law of diffusion states that particles move from an area of more concentrated particles to an area where they are less concentrated. Uh, this means that particles are diffusing down their concentration gradient. And eventually the particles will reach dynamic equilibrium where there's no change in concentration uh, in different locations in a solution. Okay? In this lab, we are going to observe the diffusion of food coloring in water and determine how temperature affects the rate of diffusion. Okay, you'll take two beakers of the same size, fill one beaker with room temperature water, and fill the other beaker with an equal amount of chilled water. And to both beakers, you're going to add a few drops of food coloring without disturbing the water, and note the time. And then you'll check the beakers at various time intervals to see uh, the rate of diffusion of the food coloring in both beakers, and you'll note your observations. So here is a picture of diffusion of food coloring in beakers. This is actual results from my past students. And if this was an in-person lab, we use hot water and room temperature water, but for the online class, you use chilled water and room temperature water. Anyway, in this picture on the left beaker contains hot water and the right beaker contains room temperature water, and a couple drops of food coloring were added to both beakers. In the middle panel, these are the beakers after 1 minute, and you can see the food coloring that was put in the hot water is almost completely dissolved, diffused throughout the beaker, whereas on the beaker on the right with the room temperature water, you can see it kind of settle to the bottom, and some of it is diffusing upward into the solution. And then pictured on the right is after 8 minutes, the food coloring in the beaker of hot water is completely dissolved, diffused, and the room temperature water, it's still, still diffusing. You can see more of the food coloring has dissolved than at the 1 minute time interval, but still it's not as diffused as the hot water beaker. All right. So diffusion in the hot water was much faster than in the room temperature water due to the molecules moving at a much faster rate. So diffusion is affected by temperature. The higher the temperature, the higher the rate of diffusion. All right, next we have diffusion across a permeable membrane. Pictured on the left, we have a high concentration of particles on the left side of the membrane, and the membrane has pores. And over time, the particles are diffusing through the pores, and then eventually the concentration of particles on both sides of the membrane will be the same. So we call that equilibrium. It's actually dynamic equilibrium because particles are still moving across the membrane, but the particles that are moving from left to right equals the particles that are moving from right to left across the membrane. So that's dynamic equilibrium. And here's another picture with two different kinds of particles. The green particles are on the left side of the membrane, and the purple particles are on the right side of the membrane. And they diffuse independently. The purple particles will diffuse from right to left, and the green particles will diffuse from left to right, and they'll continue diffusing until there's equal amount of green particles on both sides of the membrane and equal amount of purple particles on both sides of the membrane. So diffusion across the cell membrane does not require energy, so it's called passive transport. And diffusion down the concentration gradient is the sole means by which oxygen enters your cell and carbon dioxide passes out of your cells. So when you breathe in, the air that you breathe in has a high concentration of oxygen relative to the cells in your lungs. So the oxygen diffuses into your blood cells, and the concentration of carbon dioxide in your blood cells is higher than the air that you're breathing in. So carbon dioxide will diffuse out of your cells. So when you exhale, you're exhaling the carbon dioxide waste that you're generating. Okay, so that's an example of passive transport. Okay, so activity 2 is observing diffusion through a permeable membrane using dialysis tubing. If this was an in-person class, this is what you guys would be doing. But being that this is an online class, I'll give you a different exercise, but you still need to know the procedures for the dialysis tubing exercise. What you would do is you would cut a 16-centimeter length of tubing, dialysis tubing, and tie one end in a knot, and then you'd put 10 mils of starch solution in the tube, and then you would stick that dialysis tubing into a beaker containing water and 1 milliliter of Lugol solution, and you would observe the changes over time. And then in another dialysis tube, you would tie an end and put 10 mils of water with 3 drops of this chemical called phenolphthalein. So phenolphthalein is a pH indicator that turns pink in the presence of base. And what you'll do is you'll take that bag of containing water and phenolphthalein and you'll put it into a beaker containing sodium hydroxide, which is a very strong base, and you would observe the changes. So pictured here is the results from students from a previous class of mine. Pictured on the right is a bag, dialysis bag containing a starch solution, and it was placed into a beaker containing Lugol solution, and the contents of the bag turned dark, deep blue, almost black, whereas the contents of the beaker remained this amber color. Okay? And on the left, this dialysis bag contained phenolphthalein, and the contents of the beaker contained sodium hydroxide, and the contents of the bag turned a bright pink color, whereas the contents in the beaker hardly changed any color. So since the contents of the dialysis bag turned dark blue, almost black, and the contents of the beaker remained the same amber color, we can conclude that the starch was unable to pass through the dialysis tubing membrane because it was too large, whereas the Lugol's reagent, the molecules were small enough to pass through the pores in the dialysis membrane and cause a color change to a dark blue or black color. Okay. And for the dialysis tubing filled with phenolphthalein, because the contents in the beaker did not change, we can conclude that the phenolphthalein molecules are too large to pass through the dialysis tubing membrane, but the sodium hydroxide molecules were small enough to pass through the membrane into the tube and cause the contents to turn a bright pink. All right. Now, for you guys, for the online class, Dialysis tubing is not something you can buy in your local grocery store, and even if you could, it's very expensive. And phenolphthalein is also not something you cannot buy in your grocery store. But what you can do is you can buy cornstarch and iodine and plastic wrap. So for you guys, you'll use plastic wrap as your permeable membrane. So here's some plastic wrap. What you'll do is you'll make a ring with your thumb and index finger like so, and you'll take a piece of plastic wrap, place it over your thumb and index finger, and you'll push the plastic wrap through, making a little pouch like so. And then you'll fill this pouch with a cornstarch solution. And then you'll stick it into a beaker of iodine and water, pictured here on the right. And then you'll observe the change, and you'll be able to determine which molecule diffuses in which direction. Okay? Okay, next we'll talk about osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. If a membrane is permeable to water but not to a solute, and it separates the two solutions, then the water will move from areas of low solute concentration through the membrane to areas of high solute concentration. Okay, so here I have a picture. Pictured here, we have a U-tube, and at the bottom of the tube, we have a selectively permeable membrane that allows water molecules to pass through, but solutes cannot pass through. And on the right side, we have a high concentration of dissolved solutes. And on the left side of the U-tube, we have a low concentration of solutes. And what will happen is water will move through the membrane from low concentration to high concentration. And it will keep moving until the concentration is equal on both sides of the membrane. So the level of liquid on the left side of the tube will drop, whereas the level of liquid on the right side of the tube will rise. Okay. All right, so there's three types of solutions based on concentration. We have the isotonic solution. This is a solution that, relative to cells, the concentration of dissolved particles is the same in the solution versus the concentration of particles within the cell. Okay, we call that isotonic, where both levels are equal. Okay, hypotonic solution has a very low concentration of dissolved particles relative to cells. Okay, so when there is a gradient such as that, water will diffuse into the cell and the cell will expand. Okay? Hypo means low level, so hypotonic means a low amount of dissolved solutes. And finally, we have hypertonic. Hyper means high level. If you're hyperactive, you have a high level of activity. If you have hypertension, you have high blood pressure. Well, a hypertonic solution has a high level of dissolved solutes relative to cells, so water will move out of the cells and the cells will shrink. Cell walls of plant cells, prokaryotes, and fungi, they have a thick cell wall, so they hold up very well in hypotonic solutions, because as water flows into the cell, instead of the cell swelling, it will and bursting, the cell wall will protect it and prevent it from bursting because it's made up of very sturdy polysaccharides. Okay, so here is a picture of an animal cell in a hypotonic solution. Hypo means very low level of dissolved solutes, so water will move into the cell and the cell will swell and it will eventually burst. In a plant cell, the cell wall prevents the cell from bursting. Okay, so plants love hypotonic solutions. They can handle it, but animal cells cannot. In the middle panel, we have an isotonic solution. So here we have an animal cell in an isotonic solution. This equal amount of water that diffuses in also diffuses out. So there's no net change of water flowing into or out of the cell, and the cell is perfectly happy. In a hypertonic solution, we have a very high level of solutes in the solution, so water will move out of the cell into the solution, and the cell will shrivel up. Okay, in a plant cell, water will diffuse out of the cell, and the cell membrane will shrink, and we call that plasmolysis, and the plant will wilt because it's losing water. Okay, so in this experiment, we will use potatoes because potatoes, their cell membranes are permeable to water, and we will observe the flow of water into or out of the potato cells. Okay, so you'll take a potato, cut a couple of French fry size pieces. Make sure the pieces are about the same size, and observe the color and firmness of the potato pieces. And you'll have a cup of regular water, and you'll put one potato piece into that cup of water, and then you'll have another cup filled with the same amount of water, but with 3 or 4 tablespoons of salt. And you'll put the other piece of potato in that salt water. That's the hypertonic solution. And you'll let those pieces of potato sit in those cups for 2 or 3 hours. And then you'll observe what happens to the potato. So here are the results from students from a past class of mine. The piece of potato that was soaking in water is on the left, and the piece of potato that was soaking in salt water is on the right. And the potato that was soaking in water remained firm, and it's a nice light color. And when they weighed it, it actually weighed more. So water flowed into the potato cells. And the potato on the right that was soaking in the salt water, it turned out to be very flimsy, and I don't know if you can see it in this picture, but it also turned a brownish color. And when they weighed it, it actually lost weight because water flowed out of the cells due to osmosis. Okay, and this is an experiment that you guys can do at home. You'll have to buy some potatoes and just cut a few slices, and the rest of the potato you can cook and eat. So that's the osmosis experiment. Next, we'll talk about enzymes. Enzymes function as biological catalysts. They increase the rate of reaction without being consumed by the reaction, and they are usually proteins. Okay? So enzymes speed up reactions by lowering the activation energy needed for a reaction to begin. And so an enzyme is very selective in which reaction it catalyzes, and its shape determines the enzyme's specificity. Okay? A substrate is a substance that the enzyme acts upon, and the enzyme can only act on one substrate. Okay? For instance, the enzyme sucrase can only act upon sucrose, nothing else. Okay? So there is an active site on the enzyme that a substrate will fit into, and that will enable the enzyme to increase the rate of reaction Here is an example. So here is the enzyme sucrase. It can only act upon the substrate sucrose. Here is the active site where the sucrose can bind to the enzyme sucrase, forming an enzyme substrate complex. And this enzyme cleaves the glycosidic linkage between the two monosaccharides, and then it releases fructose and galactose. And then the enzyme is unaffected, so now it can bind to another molecule of sucrose, break the glycosidic bond, release the monomers, and then it will bind to another substrate, and it will continue doing so until there's no more substrate left. Okay, so that is sucrase. It can only bind to sucrose, not anything else, not lactose or fructose or maltose, only sucrose. All right. Okay, so the enzyme that we will be studying is called catalase. So catalase is an enzyme that is present in animal and plant cells, even some bacteria. And what it does is it breaks down hydrogen peroxide into water and oxygen gas. Okay, so here's the reaction. 2 molecules of hydrogen peroxide plus the catalase enzyme yields 2 molecules of water and oxygen gas. And you will need to use potatoes. Potatoes contain lots of catalase enzymes. You'll need some food coloring, some dish soap, and what we'll do is we will determine the effect of vinegar on the catalase enzyme. And you'll also need some hydrogen peroxide for the catalase to convert to oxygen and water. Okay, so first you'll cut a small piece of potato and put it into a bowl of vinegar, and let it soak there for an hour or two. And then you'll fill 1 cup with 3% hydrogen peroxide, and then you'll add a couple drops of food coloring and a couple drops of dish detergent. Okay, the dish detergent will will allow the formation of bubbles. Okay, and then you'll gently mix the contents by swirling. And then you'll cut another fresh piece of potato, equal in size to the size that you placed in the vinegar, and you'll place it in the tube or cup with peroxide, food coloring, and dish soap. And you'll observe the amount of bubbles formed, and then you'll repeat the procedure. You'll have a cup of peroxide, food coloring, and dish soap, and you'll add the piece of potato that was soaking in the vinegar. Okay, and then you'll observe the amount of bubbles formed. Okay, and the bubbles forming is the oxygen gas that the catalase is creating by breaking down the hydrogen peroxide. Okay. Okay, so here we have a picture of the results from my students from a past class. On the left is a tube containing hydrogen peroxide, some dish soap, and it looks like blue food coloring, and they put a fresh piece of potato in it. On the left is a tube with hydrogen peroxide, dish soap, and food coloring, but they put the piece of potato that was soaking in vinegar into that tube. Okay. And in the middle tube, if you're doing an in-person class, you'd also have a piece of potato soaking in baking soda. Okay. And if you notice, the amount of foam produced in the fresh piece of potato and the piece of potato that was soaking in baking soda, they produced a lot of foam, whereas the piece of potato that was soaking in vinegar, it produced hardly any foam. Okay? So what can we conclude? Well, we can conclude that the vinegar destroyed or denatured the catalase enzyme, preventing it from converting the hydrogen peroxide into water and oxygen, compared to the normal piece of potato. And it also looks like baking soda did not have any effect on the catalase enzyme because it also produced a lot of foam. Okay. Now, for you guys, you're only going to test vinegar versus a fresh piece. Here is a picture from one of my students who did this experiment online. They took a piece of potato and a fresh piece and put it into a cup containing hydrogen peroxide, and a piece of potato that was soaking in vinegar. They put that into a cup of hydrogen peroxide, and over time, the fresh piece produced a lot of foam, whereas the vinegar piece did not produce any foam. So the vinegar denatured the catalase enzyme, preventing it from functioning. Okay, and that is it. Thank you guys for attending, and I will see you guys next time. Take care.